All right, guys, welcome to 2.4, Plasma Membrane Structure and Function, the last section of Chapter 2. Let's jump into it. Okay, so the plasma membrane is also known as the cell membrane, is the boundary layer of a cell, and it's going to separate the cell's internal environment from its surroundings, right? Basically, all of the interactions between the cell and the outside environment is regulated by the plasma membrane. It includes things like oxygen and carbon dioxide moving in and out of the cell, pH, salt ions, any sort of waste, any also glucose and nutrients, right? And you can see that it's got a very complex structure, which we will have a look at in a second, but this is the inside of the cell, and then here is the outside of the cell. Now, Jonathan Singer and Garth Nicholson first proposed this fluid mosaic model of the plasma membrane in 1972. It basically proposes that the plasma membrane is composed of two phospholipid layers forming a main boundary component, which is called the fluid component, and then within that, embedded is molecules of proteins, carbohydrates, and cholesterol forming the mosaic component, and the two of them put together means the fluid mosaic, right? And so here's the main boundary layer, the yellow that's there, and then embedded in them is the kind of these proteins, these sugars, and then the cholesterol. Okay, let's start with the phospholipid bilayer. Okay, the phospholipid bilayer is the main boundary component of the plasma membrane. It is made up of phospholipid molecules, right? And these molecules, this is what they look like here, right? They have a phosphate head, which is polar and hydrophilic, meaning that it loves water. Hydro is referring to water. Philia is referring to love, so it loves water. And it contains a fatty acid tail, right? And these fatty acid tails are non-polar and what we call hydrophobic and hates water. Phobia is a fear. So these polar molecules are only attracted to other polar molecules, whereas these non-polar molecules are only attracted to non-polar molecules. So when you sprawl them out together, the heads are attracted to each other and form this blanket here. The tails are attracted to each other. And then the second layer, phospholipid layer, is kind of upside down from the first one, right? And so the tails are attached to each other there. So all of that, chemically speaking, is attracted and bonding to each other, form this kind of fluid membrane that you see, okay? Other examples of polar and non-polar molecules include water as a polar molecule. Water is polar because hydrogen is more positive on this end, whereas the oxygen is more negative on this end. And non-polar means it's balanced, right? The structure is arranged in such a way that the polar heads are attracted to each other and therefore they face outwards, but the non-polar tails are attracted to other tails and face inwards, right? This is going to prevent water from freely passing through the phospholipid bilayer, which is a good thing. You don't really want your cells to be filled with water the moment you jump into a pool, right? So it's impermeable to water, but it's also impermeable to other things that are water soluble, like ions and polar molecules, and it retains that viscous fluid structure depending on the amount of saturated or unsaturated fatty acids inside the phospholipid molecules, right? The higher the percentage of unsaturated fatty acids, the more fluid it is, right? So here is a diagram of two plasma membranes. This one here has more unsaturated fatty acid cells. You can see those kinks making it a lot more flexible and bendy, whereas this one is packed and saturated and therefore a lot more firm and closer together, right? This is an animation of the plasma membrane in action, and you can see things just kind of bounce off of it, and they won't allow everything through. In fact, they have to go through these little protein channels, right? And that fluidity that's represented here, you can see there's your red blood cells need to be able to squash as they travel through your capillaries, depending on the amount of water available in your blood capillaries. Now, cholesterol has a bit of a bad rep in the food world, but actually, biologically speaking, cholesterol is a really important molecule because it is a fatty molecule that stabilizes the phospholipid bilayer, right? It composes about 30% of your plasma membrane. At high temperatures, it's going to raise the melting point. At low temperatures, it actually packs it together and makes it more firm. The second component that is vital to both the structure and the function of the plasma membrane is the proteins. Okay, they are embedded in the phospholipid bilayer, and different proteins perform different types of functions. Some of them are responsible for transport, which is what the rest of this section looks at. Some of them are responsible for enzyme activities, breaking things down. Some of them are important for a signal transduction, which is really important for your body's ability to receive and send signals, like, for example, in hormones. And then also, some of them are important for cell-to-cell recognition. So, for example, your immune system, cells in the immune system need to recognize one another as being yourself and not a foreign body, so that your body doesn't eat itself when trying to defend from bacterial infections and so forth. Okay, cells can join to one another using these proteins, or they can join to the cytoskeleton within the cell using these proteins as attachment sites. Here's an overview of all of the different types of proteins. So, for example, this one is used for transporting ions. This one is a protein pump, transporting proteins. This one is an aquaporin, transporting water. And then here's another one as well. Okay. Now, depending on the structure of the protein, it has a slightly different name. So, integral proteins are proteins that are permanently embedded in the phospholipid bilayer. So, when integral proteins span from both layers of the phospholipid bilayer, we also call them transmembrane proteins. So, for example, here is a integral protein that cuts through the whole thing and is considered a transmembrane protein. Here's one that only cuts halfway through and is considered not a transmembrane protein. Okay, the word integral actually means integrated, which is woven into the plasma membrane. Okay, and it's permanently embedded. If it's not permanently embedded, we call it a peripheral. Peripheral meaning side vision, peripheral vision, so off to the side. So peripheral proteins are only temporarily attached to the phospholipid. They're either attached to the surface of the phospholipid, much like this one here, or it's attached to an integral protein, like this one here, and will assist in their particular function. Okay. The third component, kind of lesser known compared to the last two, are carbohydrates. Carbohydrates often attach to either proteins or lipids, and they form larger complexes and serve a variety of roles, such as adhesion, cell identity, recognition of antibodies, hormones, and viruses. If it's attached to a protein, we call it a glycoprotein. Glyco referring to sugar or carb, and protein referring to the protein. If it's attached to a lipid, it's called a glycolipid, right? So there's your glycoprotein, and there's your sugar molecule. There's the original protein. That's probably an integral protein. And if it's a glycolipid, it's attached to the lipid phospholipid molecule that's on the plasma membrane. Okay. Now, an example of the usefulness of carbohydrates in your plasma membrane is through what we call the MHC complexes, right? So MHC stands for major histocompatibility complex, and these molecules are really large glycoproteins that your immune system uses to figure out which cells belong to it and is considered as self, and which cells are not belonging to you and considered as foreign cells, right? So, for example, right, this is a natural killer cell. This is one of your white blood cells, and it's going to come along, and this is one of your cells, right? And your normal cells has a particular molecule here called the MHC1 complex, right? And basically, when the white blood cells come along, and they've got this little attachment arm, and it's going to attach to the MHC complex here, and if it attaches, it triggers and basically tells your white blood cell, hold on, this is me, and don't kill me because I am you and we are the same organism, right? If there is no MHC molecule attached on, say, for example, an infected cell or a bacterial cell, it's going to trigger a different response in your white blood cell, releasing these enzymes that come in and attack and kill this non-self cell, right? So that's how carbohydrates play a really important role on your cell surface. Okay. Now, I want to talk about transport, because transport composes the main section of 2.4. And so one of the main functions of the plasma membrane is being able to regulate the exchange of materials between the cell and its environment. In order to do this, the plasma membrane needs to be semi-permeable, right? Semi-permeable or selectively permeable means that the plasma membrane will allow some types of molecules to cross freely while restricting the movement of other types, right? That's why it's called semi-permeable, right? So if something is permeable, it lets things through. If it's semi-permeable, it will let some things through. If it is non-permeable, it won't let anything through, okay? So depending on the type of molecule, you're going to get different levels of permeability. So if the type of molecule is small and uncharged, like for example, oxygen and carbon dioxide, it's going to go straight through the membrane, right? If it is lipid soluble or non-polar, like for example, alcohol or steroids or chloroform, that is also permeable. That's going to go straight through the phospholipid bilayer, right? If it is small but it is polar, right? Remember that polar quality being related to water and water solubility, like for example, water and urea, that is going to only be semi-permeable or permeable depending on the level of water that your body wants to let through. Small ions like potassium, sodium, and chloride are non-permeable, meaning they can't pass through the plasma membrane, the phospholipid bilayer, and have to go through the protein channels, right? And then anything that is large, anything that is solid, water soluble, and anything that is large and polar must go through the protein channels as well. So that includes things like glucose and amino acids, right? Any molecule that isn't going to pass through the phospholipid bilayer must go through the protein channels. Okay, here is an overview of those different ways, and we're going to cover 5 different ways that things go in and out of your plasma membrane. Most of them are kind of related. Okay, simple diffusion, facilitated diffusion, osmosis, which is a special type of transport of water, active transport, and bulk transport, which comprises endo and exocytosis. Okay, different molecules will cross using these different methods, and the methods are either classed as transport requiring energy in the form of ATP in order to occur, or they are passive transport. Passive meaning they don't require energy. Okay, so of the active and passive transports, it's broken up like this, right? Passive includes normal diffusion, osmosis, and facilitated diffusion, steps 1, 2, and 3, and then active transport is number 4 and number 5, right, which is up here, right? All right, let's start with simple diffusion. So simple diffusion, or simply just diffusion, is when particles in a solution are going to move from high concentration to low concentration. Literally, diffusion is the spreading out of molecules, okay? General movement of diffusion is actually very slow and random, okay? So it randomly moves around until it spreads out and evenly distributes itself across the whole area. And this particular mode of diffusion between cells is passive. It doesn't require energy to occur. And when things are passive, we refer to them moving down a concentration gradient, right? So for example, it might start here, very concentrated, and then it will slowly spread to areas of low concentration, like, for example, the other parts of the environment. So it starts in the middle, and then it's going to spread until eventually it's fully spread out and evenly distributed across the whole area, okay? Now, this occurs with small uncharged molecules like oxygen and carbon dioxide, as well as lipid-soluble molecules such as alcohol, chloroform, and steroids, right? Generally, what you notice over time is this, right? Here's your plasma membrane. Here is outside of the cell, like the extracellular fluid. So for example, let's say, for example, you drink a bottle of alcohol, it goes into your stomach, and then here are the cells of your stomach, and the alcohol is going to diffuse outside of your cell and eventually make its way inside of your cell, whether you like it or not, and gets absorbed straight into your bloodstream over time until it is evenly distributed, right? This movement is random, but because molecules hit and collide and push off each other all the time, that means they are going to bounce and distribute themselves evenly, and you get drunk, right? That's why you absorb alcohol so quickly, and you can get drunk really quickly the moment you start drinking, and it stays in your blood really well. Now, the second type of diffusion is called facilitated diffusion. Now, facilitated diffusion is still diffusion, but it's been aided by a protein, right? The protein allows a means for larger molecules or charged molecules that couldn't pass through the phospholipid bilayer to then cross through these particular types of proteins, right? It's still passive in the sense that it doesn't require energy and therefore moves down the concentration gradient. It's more rapid than simple diffusion, but it actually can get saturated when all of the protein channels are used up, okay? So the membrane proteins are specific for particular types of molecules, right? So for example, right, simple diffusion goes through the plasma membrane here, the phospholipid bilayer, but facilitated diffusion must travel through these gateways, right? These channels, right? It's either a channel protein or a carrier protein, right? And the difference between them looks a little bit like this, right? The channel protein is open the whole time, right? However, it's filled with a little bit of water, which means you can't travel through it unless you dissolve into the water and pass through that water and get into the other side. On the other hand, the carrier protein, right, when the molecule attaches to it, it changes shape and allows the molecule to pass through, right? And so that's the difference between the channel protein and the carrier protein, right? Here's another diagram to kind of give you a better idea, right? So channel proteins, like for example, an ion channel contains these pores that are filled with water, allows the water to go through. It doesn't bind to that molecule, but the carrier protein does, right? When the protein attaches to that particular molecule, it changes shape, pushes the molecule through, and then once the molecule is released, the shape reverts back to its original shape, allowing the next molecule to transport through, right? All of that doesn't take any form of energy whatsoever. It happens naturally, but can slow down as a result of it, okay? So the rates of diffusion, that's going to differ between simple diffusion and facilitated, right? It depends on three factors, okay? Number one, the concentration gradient. The bigger the difference, the more of it's going to move through, right? So in other words, if it's super dense outside of the cell, that's going to push more of it inside the cell. If there's not much of a difference between outside and in, it's going to slowly make its way in. If it's even proportions on both sides, so let's say, for example, you've got the same amount of molecules on the outside of the cell as the inside of the cell, it's actually going to just move in and out at the same rate, and therefore the concentration isn't going to change at all, and it becomes even and balanced, okay? The rate of diffusion also depends on temperature. In other words, the higher the temperature, the faster the molecules are going to move and the faster the rate of exchange is going to be, okay? The particle size also plays a role. Smaller particles tend to diffuse faster than larger molecules, okay? In addition, the rates are going to differ at various times, right? As you increase in concentration, right, actually facilitated diffusion slows down. So in the beginning, facilitated diffusion is actually really fast. It's much faster than simple diffusion through those protein channels, right? But as you move on, it's going to then slow down, because what happens is in facilitated diffusion, you get this thing called saturation occurring. Saturation is when those protein channels or those carrier proteins are fully occupied. In other words, there's no space for molecules to go through, and they have to line up, wait for other molecules to transport before they can move on, right? That saturation, they can also experience competitive inhibition. Now, competitive inhibition is when the same protein, that same transport protein, is used by different types of molecules at the same time, right? In other words, the molecules are competing, and that competition is stopping them from using that channel, okay? Now, let's take a look at osmosis. Osmosis is a special type of transport. Osmosis is specifically the net movement of water, and only water, across a semi-permeable membrane from lesser solute concentration to higher solute concentration. Notice the wording is a little bit different, okay? Osmosis is when water moves from where there is little solutes to where there is a lot of solutes. And you would have learnt probably a long time ago now that the word solute is the material that is dissolved in the water. The solvent is the water itself, and the whole thing is called a solution, right? So if you were to imagine a beaker, and the beaker is separated by your semi-permeable membrane here, and then in your beaker are either ions or water, that interaction, that is osmosis at work, right? If, for example, you have a lot of salt in one side of the beaker, and the salt can't move in and out freely, but the water can, right? You're going to actually get water moving from where there is little salt lesser solute, right, or little sugar, depending on whatever it is, to where there is a lot of salt, right, or high solute concentration, right. So eventually the water is going to move from the left to the right, and you're going to see an imbalance of water between the two membranes. Now the water can move freely, but it's actually attracted to the salt. So it's actually going to move this way and concentrate up this end, right? Here's the same phenomena occurring, and you can see the water is going to move in and out freely, but eventually it's going to stack up on the side where there is more solute concentration. Now, that happens because water is naturally attracted to those solutes, right? And it's going to bind to them, chemically speaking, right? The semi-permeable membrane allows water to move, but it doesn't allow the solute to balance out, right? So that water is going to follow to the side that has the highest solute concentration until the ratio of water to solute is balanced, right? So in here, you can see, it's probably about, 4 molecules of water per molecule of solute, and that balances out on both sides, where initially this was unbalanced. Even though the water level was even, there is more water per solute ratio on this side compared to this side. Now, because the plasma membrane is permeable to water, and the cytosol is made up of water that contains dissolved solutes like salt or sugar, osmosis occurs naturally in your cells, right? It needs to be regulated and managed by the cell, but naturally speaking, it will move in and out of the cell quite freely, right? It will move from the high concentration of solute down the osmotic gradient to where there is low solute concentration. And so that means that water is going to move and spread out just the way that other molecules diffuse. And that process means it also takes no energy, right? So it goes down the concentration gradient and it takes no energy. Osmotic pressure is the amount of pressure that is required to keep the water levels balanced. And so the greater the difference in solute concentration, the greater the osmotic pressure is, right? Osmosis straight through the phospholipid bilayer is one of the main ways that water passes through the cells, but water can also pass through the aquaporins, right? These integral membranes proteins that serve as channels for water to pass through, right? So here is water passing through those aquaporins, allowing it to move in and out freely, and then also being able to move through the plasma membrane as well if it wants to. Water. Now, alongside that, we also refer to that concentration of solute as tonicity, okay? When you have differences between the concentration of solute inside and outside of a cell, we have different words to refer to that particular type of solution, right? If the solute concentration inside the cell is the same as outside of the cell, we call that isotonic, right? So that cell is sitting in isotonic solution. And when it's in isotonic solution, the amount of water that goes into the cell will be the same as the amount of water going out of the cell. That means you're not going to experience any change in the cell whatsoever, and that's a good thing, right? That means that it is balanced and the cell will not experience a net movement of water of any sort. And in many ways, most cells want to be in an isotonic solution, right? If the cell is in a hypertonic solution, hyper meaning high, tonic referring to tonicity, right? That means that the solute concentration is greater outside of the cell than inside. In other words, water is going to move out of the cell more so than water going in. That means water eventually leaves the cell and the cell is going to shrivel up, right? And that is not a good thing. You don't want your cells to shrivel up, right? A good example of a hypertonic solution is when, before they had fridges, they used to put meat in salt and they would salt these foods. And what the salt does is the salt is a hypertonic solution. In fact, there's literally no water outside, right? And so all of that salt is going to draw water outside of those meat cells, and that's going to dry up the meat and that's going to preserve it. because it will remove all of the water and it will kill the bacteria at the same time as well, because the water inside the bacteria gets pulled out into the salt and the bacteria shrivel up and die, right? So that is a hypertonic solution. On the other hand, a hypotonic solution, hypo meaning down, right? That means that the solute concentration is lesser outside of the cell than inside. That means the water outside of the cell is less than the water going in, and water is going to try and move into the cell, causing it to swell. And if it continues to swell, it will eventually burst, right? Also a bad thing, you don't really want your cells to burst. And so it's really important that the cell needs to maintain an isotonic kind of ratio, okay? It's important to note that cells actually cannot move water themselves, right? Cells don't actually have a way of moving water. Instead, what they actually do is they move the solute. They can pump solutes in and out of the cell, and the water follows that particular solute concentration, right? So, for example, right, if a cell is a bit flaccid, it's going to pump salt into the cell, and then the water is going to follow and then balance out that particular ratio. On the other hand, if it's turgid, you're going to get the opposite effect occurring. Okay, now let's talk about active transport. Active transport is referring to the use of energy to transport molecules through your carrier proteins, right? It is also referring to any type of transport that uses energy. So it's kind of ironic. It's both a category of its own and a broad category referring to any time you use energy, okay? Active transport only occurs through carrier proteins, right? Whereas facilitated diffusion occurs through both carrier and transfer protein as well, right? Channel protein, sorry. It uses ATP as the primary energy supply to the process, and the transport molecule goes up against the concentration gradient and uses up energy in the process, right? Similar to facilitated diffusion, active transport also experiences saturation. In other words, that same protein could be used up completely and the molecules have to wait in line, and competitive inhibition when the same protein gets used by multiple different types of molecules, right? The same molecule can be transported via facilitated diffusion and or active transport depending on the needs of the cell, okay? So here's a diagram of active transport occurring, and you're going to notice that little ATP molecule that I mentioned in the previous chapters attaching to that carrier protein, changing its shape and releasing that molecule, right? That's how ATP pays for that particular transaction. That's how your body uses molecules of ATP to transport things in and out, okay? Here's an example, right? Glucose in the gut, right? One of the key nutrients that we get from food is glucose, right? Glucose is absorbed from the intestines into the bloodstream through what we call epithelial cells, which is your intestinal linings, right? So you imagine you eat a burger and then it gets digested and goes through your intestine, and as it passes through here, right, it breaks it down into glucose, and it's going to pass through the intestinal lining and get into your blood where it gets transported around the body, okay? Now, that intestinal lining, right, here, active transport from the glucose transporter protein extracts all of the glucose from the gut against their concentration gradients, right? If the transport was passive and your body cannot actively move all of the glucose, what you're going to see is, let's just imagine if it was facilitated diffusion or whatever, right? Once the glucose here and the glucose inside the cell is the same, it's going to stop absorbing glucose from your gut, right? You don't want that, right? Because that means all of the remaining glucose, you're going to poop it out and it's going to be wasted, right? So what happens is, even though that ratio moves through naturally and it balances out halfway between what is in your gut and what is in your intestinal lining cells, your epithelial cells is going to then use active transport to pull the rest of the glucose across the membrane and absorb all of it, leaving none in your gut. That means that it is going to be low concentration here, high concentration here, but it's going to keep moving up the concentration gradient from low to high until eventually there is none left, okay? The glucose will then move into the capillaries via facilitated diffusion in this particular process. All right. Another example of active transport occurs in your sodium-potassium pumps, right? Now, sodium-potassium pumps are super important. Why? Because your nerve cells use these to transmit signals, right? Literally, for you to think, for you to contract your muscles, for you to move, for you to function and send a signal to your organs to digest your food in your stomach, for example, you need your nerve cells to do this particular function, okay? So nerve cells use sodium, right, Na plus and potassium ions, K plus, to transmit signals, right? These signals are transmitted by releasing potassium out of the cell, and sodium goes into the cell via these ion channels, right? This happens via facilitated diffusion, and it doesn't require any energy. However, that little mechanism of releasing potassium out of the cell and bringing in sodium into the cell, that needs to be reset in order for you to send another signal, right? So your body needs to find a way of bringing back the potassium into the cell and then pushing the sodium out of the cell. And to do that, it needs to use sodium-potassium ion pumps and active transport to make it happen, okay? So how it works is sodium-potassium ion pumps are carrier proteins. This is what they look like there, right? And each carrier protein binds to 3 sodium and 2 potassium, right? 3 sodium ions inside the cell binds to the carrier protein, and then one molecule of ATP comes along, attaches to it, and this releases just enough energy to change the shape, and it releases those 3 sodium ions outside of the cell. And then that process is called phosphorylation. And then what happens is 2 potassium ions then bind into the carrier protein at the same time. And then that's going to change the shape of the carrier protein, releasing them back into the cell, okay? When your nerve then transmits a signal, that potassium gets released out of the cell, that sodium goes into the cell, and then it sends a signal down the axon of your nerve, right? Allowing you to send a nerve signal from one nerve cell to the next nerve cell and move that forward, okay? Now, the resetting of that mechanism is where the energy is required. And so when you notice when living things die, after a certain period of time, their cells begin to break down, and actually the sodium gets released into the cell, and the organism, because it is dead, no longer has a way of resetting the mechanism because it doesn't use energy anymore. And so what happens is your muscles begin to actually all contract at the same time because of this release of sodium and potassium all around the muscle cells, right? And so that process, that's actually called rigor mortis, when an organism becomes really stiff and firm because the muscles are all contracted, and there is no way for it to reset the mechanism and kind of soften once again. Okay, bulk transport. So bulk transport is another type of active transport, and it is the last of the transport methods that we're going to look at. And it is looking at moving large molecules or fluids either into or out of the cell, right? It involves 2 processes, either endocytosis or exocytosis, okay? Endocytosis is movement of substances into the cell, right? Endo meaning into, cytosis is the cell, so moving things into the cell. It occurs by the pinching of the plasma membrane to form a vesicle, much like how your Golgi apparatus back in 2.3 does it as well, okay? There are 3 types of endocytosis. Pinocytosis is when you move fluids and nutrients such as proteins and sugars into the cell. So there it is there. It pinches, it forms this little vesicle, goes into the cell, gets used by the cell, right? Phagocytosis is the entry of large particles like bacteria and cell debris, right? This usually occurs in something like white blood cells for the purpose of immune defense, right? Here is an example of a white blood cell just eating a piece of bacteria that would have sucked it in via endocytosis or phagocytosis and absorbed it. Here is the white blood cell eating the bacteria, and the bacteria realizing it's getting eaten and going absolutely nuts. Sorry, this is an amoeba, not a white blood cell. Okay. And then there's also a particular type of endocytosis called receptor-mediated endocytosis, which is a way in which your body absorbs a particular molecule like a hormone through receptors and takes it into the body to be used by the body. Okay. The other type of bulk transport is exocytosis, movement of substances out of the cell. Exo meaning out, cytosis is movement of the cell. Transport vessels containing either waste or secretory substances will fuse with the plasma membrane, releasing the contents out of the cell, or enzymes and hormones and waste products can include, excess amino acids, right? All of those kind of getting dumped by the cell out of the cell and pushed out of the cell. It also includes, for example, let's say, you eat a meal and the food goes into your stomach. Turns out your stomach releases a lot of enzymes that actually is produced by another organ called your liver. And those enzymes, those hormones and things like that, all of that needs to be produced in one spot, but then moved to a different spot from your Golgi apparatus via your plasma membrane. Okay. Now, there are two types of exocytosis, either regulated, which uses specific secretory vessels and regulated by a particular signal, or unregulated, right? Which basically, as the name suggests, occurs without the signal, okay? So here is regulated exocytosis, which is regulated by a signal from the outside. And then here is unregulated, where your body is simply getting rid of stuff. Whereas this one is probably, triggered by a hormone of some sort telling you to release a particular type of enzyme or a particular type of protein. Okay. All right. I think that's it for 2.4. I hope you guys enjoyed that, and I will see you guys in chapter 3.